Good morning. I'm Robert LeBoyer, the Senior Biotechnology Analyst at Noble Capital. With us as our next presenter is Elon Hadar, the CEO of Silexion, a company in oncogenes and gene silencing and expression. Take it away, Elon.
Thank you very much. So, good morning all. Nice to see you. You know some of the faces. So I will try to convince you that we know what we do, what we are doing. We know that we have a drug that can work. We know how to manufacture it, but we only need to get the cash. And that's probably the most challenging thing these days. So as a public company, I need to show you the forward-looking statement. You can read it after if you wish. So what we do? So we are in the KRAS-focused RNA treatment. We are trying to cure the cancer before it happens. You can never cure the cause of the cancer. You can never cure the mutation in the DNA, but you can try to stop the cancer from growing right after it happened. That's exactly what we are trying to do. So the company developed sRNA technology back in 2008. Technology came out from Hadassah Medical Center in Jerusalem, Israel. And the company went and developed the first generation of their product, and that product went through a phase 2, and I represent the phase 2. And I came to the company back in April 2022, after the recent CEO had a health issue, and I took over the company, and we changed the technology, we changed the product, and also took the company public with my CFO that is sitting right here, Mirit. We did it back in April of 2022. So, if you are not in the terms, just as we said, that there is what is known today as the KRAS oncogene mutation that is causing many types of cancer. And the company, Silexion, decided to initiate with pancreatic cancer. That's the first type that we went with. And we are covering with the KRAS-D, the KRAS-V, over 70% of those types of cancer caused by the mutation, and we are trying to treat that. But the KRAS is also responsible for other types of cancer, as you can see here, colorectomy, and other that we will address probably in the near future as well. So how do we do that? So the pancreatic cancer is really the worst that can be out there. 14 months in average, that's the expected life expectancy of those patients. And as you can see in the slide, you have really three types of the cancer. You have what we call the locally advanced, and the metastatic. So the metastatic is probably the end, when the cancer will go to the liver and to the rest of the body. But we try to focus over here on what we call the locally advanced and the border resectable pancreatic cancer. And those patients, if you can save them, that will be a home run, of course, for the company. So how do we do that? As I said before, when the cancer emerges in the DNA, the KRAS mutation, you cannot cure that. And then what happens is that the body will get the wrong signal from the mutation to grow more cells. We are trying to stop it here at the source. We are trying to put our sRNA into the tumor and stop the body from growing the tumor by getting the right signal from the protein, from the cells, and not taking the mutation that is causing the body to grow the cancer. That's basically our technology. I will not go too much into it, probably where most of us are not scientists in, but that's what we do. We try to stop the cancer before it grows. We don't try to kill it, we don't try to take it out, we try to stop it from growing. So we know that treating with sRNA is really safe, and other drugs try to do the same with small molecules. They did met many challenges while doing that, but we know that treating with sRNA is really safe. It is safe, the body tolerates the treatment, and you can really put sRNA into the body, and you can hope that the sRNA will go in the body into other areas as well, like the liver, kidneys, and will stay there, because that we can also stop the cancer from moving around. That's also important to say. So I don't need to convince you that it's a huge market. If we can find a solution to pancreatic cancer, the numbers are enormous. It's over 2 billion in the US, Europe 3 billion. So it will be a home run for sure. So the company developed the LODER. The LODER went into phase 2 trial. The trial began back in 2018, and we finalized the trial back in 2023. The trial had 2 cohorts. One was LODER plus standard of care, and the second one was only standard of care. And the second cohort was only the LODER with the standard of care. There was no what we call placebo. So the placebo standard of care, but only that. And the results from that trial really gave us the feeling that we have something. That we have something, we can extend the life of those patients, and you can see some statistics from the trial. You can see in the US, 5 sites, in Israel, 5 sites in Israel, 4 sites in the US. You can see some demographics, the age, and the cases of G12D and V mutation that went through the trial. Jumping into the results, this is change, as you can see, that's a change in percentage, it's not percent, so 0 minus 0.2 is 20%. And each bar is a patient. Each bar represents a patient and what happened to the tumor size going through the trial. So as you can see, some patients, the tumor did not shrink, but most of the patients going through the trial, the tumor did shrink. And when it says R, the meaning is that that tumor was resectable and was taken out. And taking out the tumor meaning that patient will really extend his life expectancy. And you can also see the response with chemo and chemo alone, and how the chances improved of those patients. So that was cohort 1. Moving to cohort 2, more patients, but some of them the same as well. And again, you can see that most of the patients, the tumor did shrink during the trial, and you can see again the R's, meaning that those patients could go through surgery and the tumor could be taken out. If you can repeat those results in the phase 3 in a large study, 400 subjects, that will be a home run. That will be a huge success, really extending the life of those patients. And that's what we're trying to do. And showing you the results in total. So the LODER extended the life of the patients by approximately 9 to 10 a month, from 13, 14 months to 23. And that's what we call a success. But I must say that this trial was not statistically significant. It was never meant to be. It was a small trial. But that gave us the feeling that we have something as a proof of concept, and it's something that we want to continue pursuing and developing a product that can help other patients. Just to say that treating the body with siRNA, I think it's known today to all, it's completely safe. I'm sure we all heard about those patients that took the vaccine, Pfizer vaccine or others, for COVID, and took it again and again and again, and nothing happened to them. And we know the same. We know that treating people with siRNA, you will not see serious adverse events related to treatment. And as you can see it here, and it's all been reported, and the LODER was overall well tolerated. The LODER basically was a small wad implemented into the tumor, releasing the siRNA over time. That was the LODER, and again, completely safe. So we have decided to enhance the LODER and make it into a better product. We went and worked with the big names out there for developing the siRNA more and developing an extended release technology that will give the siRNA a place to be and to be released over 3 months in the body. So those patients will not have to come to get treated every few weeks. They can come every 3-4 months, get the treatment and continue with their life all the time that, of course, it helps. And we went and developed SIL204, that's our new generation product. That product is supposed to go into clinical trials at the beginning of 2026. We already got the approval for that. And what we did, we said that we need to improve the stability in the body of the siRNA, that the siRNA will stay more in the body, and you can see here the numbers again, going up from 1 hour to 48 hours, over 48 hours stability. We went from those PHA deposit rods to microparticles, and it's really small microparticles, you cannot see them with your eyes, but they're all in a solution injected into the tumor, and the microparticles are being manufactured by Evonik, the chemistry company located, the mother company is located in Germany, and we are working with them. And we will have to show, of course, efficacy when we go into the trial. And as I said, we already have the approval to initiate a human trial. Now we'll discuss that in a bit. So giving you some data regarding SL204 and what we did, and you can see here that the product is much more stable. Though there was stable only for less than an hour, basically in the body, and now we can see that SIL204 is stable for over 48 hours and stays in the body after it is released. That's really a good sign for us. And also when we put it into rats to check stability, we don't have yet the human data, but you can see that the blue bar over here, you can see that the blue bar over here is the highest with our competitors that are also using RNA to treat tumors, and our product is much more stable for longer in the body. And that's a good sign. So we do believe that when we will have the human data, we will have also better data, stability data in the cells. We also improved the sRNA, so we can cover more mutations, and where you see that the number is over 85 down here, percentage-wise, meaning that we can cover the mutation. So we can see that we have the wild type, what we call, where you cannot really specify the mutation. We have the D, of course, we have the V a bit less, and we have here additional KRAS Q61H and G13D, and those mutations are responsible for many types of cancer that we will be able to treat in the future after we will show that we have efficacy with our first treatment, the pancreatic cancer. So now I will present you some studies we've done in rats and others to show that the product is working. Again, we are taking human pancreatic tumor lines and we put them into rats, what we call xenograft them into rats. And you can see here, again, I'm showing many of these curves, that when the tumor are not treated, you can see that they are really growing. And when they are treated, they stop growing. They grow a bit, but they do stop growing. And you can see that it dose dependent. And that's a really good sign, showing that the product is working. And moving to the same, and showing here, again, the same, what we are doing, what happened to the tumor, that you can see that the necrosis is really growing after 15 days of treatment, and this is statistically significant. We did it in many rats, we did until today probably 30, 40 of those studies, and we get the same results again and again, showing that we are killing the tumor and that the tumors are not growing with the treatment of the siRNA. And again, same, showing that the tumors, and you can see here the necrosis in the pictures, those are real pictures taken from the cells, and the white areas, the pinkish white, that's necrosis, showing that the tumors are dying when they are dosed with the SIL204. We can repeat those again, we are repeating those studies again and again. Another one. So what we did here, and this is, we will announce that also in a conference soon, that when going with chemo and taking SIL204, we really see that chemo is not that effective treating tumors, but when we do it with SIL204, we can really show that the tumors grow much less. That the chemo is really not that effective, but with SIL204, you can see that the chemo is not that effective, and we are effective. and you can see here some picture taking of the necrosis in the cells. Same results again and again. So what is the plan? What is the plan? How we want to make this into a commercial product? First of all, we need to initiate a trial. So we met with the German authorities, discussing with them the plan. We decided to go to Europe first because we knew that in Europe they will accept the safety data that we have from the Lodel. They will not ask us to do the whole safety test again as the FDA. So we went to Europe, we met with the German authorities, and they gave us the go-ahead to initiate the trial, and we will initiate it in Europe, and then we'll come to the US. And we hope to do it at the beginning of 2026 to initiate the human trials in Europe, and as I said, to expand it to the US after. So during 2025, what we are going to do, we are going to do manufacturing of the product with our partner, with Evonik. They will manufacture for us. And when we are done with that, we will go again to the German authorities, submit the clinical trial, what we call protocol, and get the approval, and initiate the trial. That's basically the plan. And that's the design of the plan. There will be one arm, of course, of standard of care. You can call it a placebo, but it's a standard of care plus chemo. And the other arm will be SIL-204 plus the standard of care. You always need to go with standard of care. You always to provide the chemo as well. The regulatory agents will not allow you to give only SIL-204 and not to continue the standard of care to those patients. And as I said, we did get the approval for that design. Our plan is to do the locally advanced pancreatic cancer and then to try and initiate studies in colorectal cancer as well, because those cancers are also generated from KRAS mutations, as we know. And the only thing we have to do is to change the delivery system targeting those types of cancer. Some names in our scientific advisory board, and we do have the best names out there. Professor Eileen O'Reilly, she is the leading investigator in pancreatic cancer today from New York. She is really taking part in the company, and you can see the other names that believe in the product and help us in developing the product, and also they will also take part in the trial. Some names in the company. In my past, I was working for Pfizer. After I went to Formix and then Penelform, and recently I became the CEO and the chairman of Silexion. Dr. Mitchell Shirvan is not here with us today. He's our chief scientific officer. He has a massive scientific background. He's leading all the work in the company. Merita, our CFO, is here. And we have Ilan Levine. He was the CEO of Stratasys back in the days, and he's also taking part in managing the company. That's it basically for today. That's what we do, and we try. Our aim is really to bring innovative treatment to those patients, to those cancer patients, and try to improve their life quality.
Thank you.
Thank you. And I will be happy to take some questions.
We can open the floor for questions if there are any.
How do you monitor what stage of cancer you're in if you're going to claim for early intervention?
So usually when those patients, the pancreatic patients will come and the cancer will be detected, it will probably be late. It's not that we can really do anything about that. So if you get such cancer, your best chances are that it will be detected when it's still in the 10-20% and it will be resectable. But many patients, it doesn't happen. They come and it's being detected here when it starts to block the blood vessels. When it's going to the liver, really, that's really game over. So, but what we are trying to do, we are trying to focus with these patients because we know that we can really help it, because if the tumor will shrink and they will go into operation, we can really extend their life. But we are also seeing that our siRNA, when put into the body and it is stable, we can find it in other tissues. We can also find it going to the liver. And if it's going to the liver and will stay there, we hope that it will also help with the metastatic phase of the cancer and will not and will stop the cancer going everywhere. That's also something we hope to see during the trial. But right now, we are focusing in the 30-40%, and we are not trying again to locate the cancer early. Our target is when the cancer was located to stop it from growing. That's what I was trying to show here, that we are not trying to, again, to cure the cancer, we can't cure it, but we are trying to tell the body, stop growing the tumors. By saying to the body, ignore the mutation that you have, the DNA mutation basically, ignore it, take the signal we are giving you, and stop growing the cancer. That's what we are trying to do when I'm saying early. We don't want to take it out, we're not trying to do other things to it, or we're trying to stop the body from growing the tumors. Of course, after it was done.
Have you seen any pattern of the stock being affected by the unrest in the Middle East? You think it can rebound and take off when it ends?
So first of all, what is happening in the Middle East is terrible for all, and for all Israeli companies are suffering. But because of that, I made the strategic decision to take the company out of Israel. So the company now is domiciled in the Cayman Islands and is a domestic filer here in the US. And a long time, and when we can get the money and initiate the trial, most of the operations will move to the US. Because I don't see any advantage of being in Israel, located in Israel, and running trials in the US and Europe. I think you need a US-based team, and that will be successful. And we did it in the past. In the past at Formix, when we launched successfully 2 drugs in the US, initially the company moved from Israel to the US. I stayed out of a job, but that was the other part of the story, but it was a successful, it was a home run. Yeah. Yes, please.
What kind of a moat do you have vis-a-vis patents so that if successful, you can't be copied, ripped off, and et cetera?
Thank you, Steve. That's a really important question. Thank you for reminding me. So it is in the last slide, and I will just repeat that. We have IP exclusivity through December 2043 without extensions, plus extension it will be more. And we do cover the siRNA composition. We have 2 patents. And the know-how is coming from Evonik. Evonik is not willing to share their know-how how to manufacture those micro particles, but they are the leading company in the world doing that. Thank you. Yes, please.
What were your contributions at the companies you'd worked to prior to starting this?
My contribution
Yeah,
to the company, other companies?
they worked to prior.
Yeah, so at Pfizer, I was the finance director for the Israeli subsidiary at Pfizer, launching products in Israel. Then I moved to Formix. I took Formix public back in 2014, raised with the help of Mirit back in Formix $350 million or so. And as I said, we launched successfully 2 drugs. I was the country manager of Formix in Israel and the CFO of the group. And after that, I moved to a pen form. Pen form, the product was not that successful, so the company didn't do so well. And then I came to Silexion.
Thank you.
I have a question.
Sure.
Very interesting research, very interesting product and
Thank you.
mechanism. Could you just go over some of the milestones and things that are ahead for the company and things that would drive the stock?
Absolutely. So 2025 is the year where we are going to do manufacturing, GMP manufacturing. As I said at the beginning, you have 3, to be successful in developing pharmaceutical product, you need to do 3 things. You need to show efficacy, you need to know how to do manufacturing, and many companies fail in manufacturing. It's not a secret. And you need to get the money.
With more than 6,000 small and micro-cap companies listed, if you're looking for the next Apple, then Channel Check truly is your tree. It's an investor community where you can access independent Wall Street research, advanced market data, balanced news, corporate videos, and podcasts. You can even register to attend in-person Meet the Management Series luncheons, and to be part of the preeminent annual Small and Microcap Investor Conference, NobleCon. And here's the best news. Since Channel Check is sponsored by the featured companies, premium membership requires no investment. It's absolutely free. So check it out. Log in today. Find out why Channel Check is North America's leading source of research on the companies that just may change the world.